RT-PCR data usually comes in one of two formats. Tall skinny format, where each row is a single measurement from a sample with additional metadata. Here the important columns are the plate, sample, target identification such as gene name, and the data, usually threshold cycle or CT. You might also have matrix format, where each row contains all RT-PCR measurements for one gene across all samples, or vice versa, and additional metadata can be present as neighboring columns. In either case, additional metadata can be added from separate files, for example, annotation metadata such as gene information. You can also add experimental design metadata such as sample grouping and treatment levels. To ensure compatibility between RT-PCR experiments and integration with other data such as RNA-seq or microarrays, a common nomenclature of assays such as sample IDs and gene IDs, such as ensemble IDs, should be used.